So let's get started here. And it's a great pleasure to have Chris Mason from Weill Cornell with us today. As you can see, anybody who's watched these webinars over the past couple of years will notice I'm still actually in my bedroom here. So despite the fact that as a kid, I was super interested in space travel, I haven't thought about traveling too far in the last couple of years. But as we know, Chris is one of the few people thinking about a long-term survival of the human race and what it would take to get us off the planet and actually live long enough, presumably, to make it to a different solar system or whatever, and what these effects are going to have on people who leave the planet. I guess principally radiation. So Chris suggested that I just point you to his web bio because it's pretty long, but I'll give you a brief intro. He's an associate professor, I believe, of genomics, physiology, and biophysics at Weill Cornell Medicine in New York City, as well as director of the World Quant Initiative for Quantitative Prediction. Earned his PhD in genetics from Yale, where he continued to do a postdoc while also, this is a kind of a first, serving as the first visiting fellow of genomics, ethics, and law at the Information Society Project at Yale Law. He joined Weill Cornell in 2009. Now that I know you're in New York City, it makes a lot more sense because Yale is right, basically part of New York City, right? His lab is working on a 10-phase, 500-year plan for the survival of the human species, and develops and deploys new biochemical and computational methods in functional genomics, essentially to help us prepare our genetic defenses for long-term space travel. Welcome, Dr. Mason. Let us know what's cooking.
Thank you. Well, and can you hear me okay and see me okay and see the slides okay? Everyone looking good?
Yep.
All right, great. Well, thank you so much for having me. It's a pleasure to be here. And I'll walk you through in about 50 minutes or so, what is the work that we've done, what are we planning to do, and what's coming shortly. So I'll start with just some fun facts, is that more than ever, we are a spacefaring species. You kind of see here, it used to just be China and Russia. This is a plot of everything that's been launched into space since 1957, since Sputnik. And you can see it's really started to pick up. And then if you look in the past couple of years, it's gotten super exponential. So now past actually 12,000 objects launched into space. To be fair, a lot of these are actually Starlink satellites, which you can see here. So it's kind of hovering around the planet. But that just opened up internet during the Ukraine war, for example, which was great to see. So that's very exciting. We've also begun to go out towards the moon. This is from the Chang'e 4 mission, which grew cotton on Earth soil that was on the moon. But more recently, we even took lunar soil and have grown some plants. We being Rob Pearl and colleagues at NASA have done this, which is really kind of cool to see that you can take this really harsh lunar soil and even grow plants in it. And so that's led people to start to put up these architectural renderings of legit plans to go and build a moon base and moon labs and moon structures and start to actually plan for what will happen when we live on the moon. And of course, the long-term goal is to make it so we're a multi-planetary species, so we can have boots on the red planet eventually, use technology and science to enable exploration. As you can see here is a great visualization from NASA. And so with that as a backdrop, it's fascinating, but it's worth also taking a moment to think, well, who's been going to space and what are the risks to their body and how can we learn what they are and even prevent them or mitigate them? And you can see it used to be all dudes going up into space. This is the number of astronauts out to about mid-2020. And then eventually, we did start to get to more female astronauts. Some of the missions I'll tell you about today, we had full parity of half male, half female crews, which is great to see. Also, it's not just all Americans and Russians. We start to get it a lot, not just more of our chromosomes, but also from many more places. We're seeing different astronauts coming and flying up into space, which is also, again, great to see. And so with that as a backdrop, the other thing that's been, I think, really phenomenal is how fast it is that we can now sequence DNA. This is the cost to sequence one human genome over the past few decades. And you can see it used to be hundreds of millions of dollars to get one human genome sequenced. And now it can be less than $1,000. And these days, it's about $200 to get the full genetic catalog of what's in your cells that could inform medical care and treatment options. And so this is an extraordinary pace of technological change. And what this is leading to is that we're still finding literally new genes. The simple question you might have is, how many genes does the human genome have? You can see it's changed a lot in the past 10 years because we're still sequencing deeper and deeper and finding rare genes that are expressed in specific cell types. You can see we now have over 60,000 genes in the human genome. It's kind of exciting. We keep discovering them and there's more to discover. And we're not just doing sequencing for human genes. If you sequence and look in the environment and do characterization there, We're also finding new species across the phylogenetic tree. We're picking up lots of new species continually being discovered that are just there waiting for us to be found, except for birds. You can see here birds are kind of plateaued. Birds are big and easy to spot, and there's not that many of them. But you can see here for almost everything else, we're finding and cataloging an extraordinary diversity of life on this planet. And so this is true even in space. We found and published with Katsuri Venkatsuran from JPL that there's even new microbes that have evolved so much they're literally different from what the ones you would see here on Earth, which is kind of exciting, I think. And so what this means for me as a geneticist is that every day, because of this capacity, is the best day to do science, which makes it very, very exciting, I'd say. So with that as a backdrop, I'm going to walk you through what we have discovered and what we're planning on doing and what we are doing for the next, quite literally, the next 500 years, hence the name of the book, of course. So phase one is what I wrote this about 10 years ago. I've kind of sketched it out as a blog post of what I think we should do. And that I knew at the time that we were getting cheaper sequencing, but we still needed to understand what happened to humans in space and in harsh environments and start to put this together. So we were selected as one of the teams to help lead the NASA Twin Study, which is a unique opportunity where Mark Kelly was staying on Earth for a year and his brother, Scott Kelly, was going up into space for a year. So it's really kind of an extraordinary chance to also, we now have the Senatorial Genome Project because Mark Kelly recently reelected as a senator from Arizona as well, although that wasn't part of the plan. It just became an interesting side part of the study later on. So this led to the NASA Twin Study, which is us to be able to examine the dynamics of what happens to the body in space for a year. And you can see when he flew up from Kazakhstan, the rocket launched out. We got up into space, started taking these beautiful pictures. You can see here the thin atmosphere of our planet. And he also did some interesting experiments. So Scott Kelly was up there. We did blood, stool, saliva, all sorts of samples. Also did a multivalent flu vaccine. You can see here doing a self-administration, taking the needle, pulling off the protector, doing injection. It does still hurt in space. So you see, ow, there's a little bit of an ow. It is still a needle. And did a full injection, got it to work, and we were able to monitor T cells and B cells and other immunological changes after that as well. But it worked out well. We actually saw that it did well. I got great diversity. And so did all kinds of things in space for a year, including juggling. It's actually very easy to juggle in zero gravity because they stay still. Grew some flowers, did a lot of work, did a lot of science. And then after a year of time in space and collecting samples, he actually undocked from the space station and started floating back here towards the Earth and was making his way, away from the ISS. And eventually we were glad to see shoots managed to deploy up in the upper atmosphere. He managed to then kind of float through the atmosphere, get down towards the ground crew in Kazakhstan, and then eventually was aided out by everyone there on the ground, giving him smiles. And he kind of said he was moving, and he gave us a great kind of thumbs up, which is wonderful to see. But even after the thumbs up, though, there was a challenge. Walking on Earth for the first time after a year with no gravity was not easy. So you can see here, just moving around was a bit slow, a bit, unbalanced. But that was the first time. And he also had challenges where his skin felt like it was on fire. He felt like he had rashes and discoloration anywhere he had contact, and he wanted to even go to the emergency room. And so the big question that we've had and then and still a little bit now is really the why. Sort of what happened at a genetic and at a molecular level to have driven this kind of response in his body. And so we started to look basically at the expression data, cytokines. One of the first things that we could see was really big difference was these huge spikes in cytokines that are pro and anti-inflammatory cytokines that would spike up on the day that he landed that we didn't see before or after the flight, and they're certainly not in flight. So really, really striking differences here, like C-reactive protein and IL-10, IL-6. IL-6, for example, spikes when you have COVID, if anything. So it's like almost the body views it as an infection. If you look at the networks, though, it's mostly about muscle regeneration is what's occurring here, basically. So you can see that's a lot of what's occurring inside his body. But we also looked at other modalities of biology, not just cytokines, but well, what else changed, basically? So you can see here, we want to look for clusters of biology that would move in concert. So it's what goes up and down in response to spaceflight. And we look for things that would do it all at the same time. So metabolites, cognitive metrics, proteomics, cytokines, telomeres, that all went up. And one of the surprising ones is that telomeres increase in flight. We work with Susan Bailey on this to validate that, but that's now been seen in multiple missions. And we could see that there's a lot of biology of things that move up and down, but in some cases, a lot of things that also mostly go back to normal after the mission, which is kind of good to see. The things that do change, though, were genes including the immune system, DNA repair, and hypercapnia. And hypercapnia, everything about it's actually when you have too much carbon dioxide, which was kind of surprising. And Scott Kelly talked about this in his book. But really, the surprising thing was, if you think of why are we seeing high carbon dioxide, part of it is because, as exemplified here by Jack Fisher, air does not move the same way in space as it does on the ground. You can actually see that you'll get mini clouds of CO2 that can build up in front of your face if you don't have a good airflow. And this is just something that we can pick up in the blood work, which was kind of intriguing to see, as you can see here for this image. We then also looked cell by cell expression of the epitopes on the outside of cells, and we could confirm actually using this PCR handle and antibody-specific barcode, taking thousands and thousands of individual cells, that we did see natural killer cells change and monocyte cell populations did change. And keep that in mind because I'll come back to this in a little bit, but you can see that these are the cell populations that would change at both the epitope level on the top as well as at the transcript level at the bottom. We can see these cells changing their response to spaceflight. So that was kind of a quick snapshot of my favorite study from the past 10 years. We've got a lot, at least relative to space. We've got all other things we've published that are about cancer or other work that is also wonderful. But in terms of the best, sort of most significant study we've done, that was the one. But then even, 10 years ago, I thought we will eventually need more astronaut studies. We proposed this multi-omic monitoring for astronauts, which looks like this, that every layer of biology that we can possibly measure, we do. And I published this in multiple journals. But now the question is, we really don't have an N of 2 before, so could we get more astronauts? And indeed, we can. So we've been now set up an IRB where people can enroll in our study. It's been open since 2021. And we call this the SOMA, the Standard Omics Measures for Astronauts, which is a really rich, in-depth profile built off the twin study methods that gives us a high-resolution genetic and molecular map of what happens to the body in spaceflight and also when a person comes back. You can see here, these are some of the assays. I'll walk you through some of them right now. So we had the first chance of this was for the Inspiration 4 mission to launch this IRB and work with Jared Isaacman and the crew to get this going. And so actually had a wonderful opportunity to be able to see the launch. So if you've never been to one, I highly recommend it. You can actually see them loading the fuel onto the rocket. It launched up into space. You can see here, got these beautiful contrails as it was getting to the upper atmosphere. And the crew, at first, all civilian crew was going up to a higher altitude than the space station or Hubble, making its way into pretty deep space. You can see here they were all excited and smiling and happy. And they also then got to work. They did some science, did some ultrasounds, did some swabbing, and then basically the day that they went coming back to Earth after a 3-day mission, splashed down in the ocean, got picked up by the crew. They were all very excited, of course, because they made it back alive. We were all very excited, also because they made it back alive, and they all brought a lot of samples with them. And then once it got up onto the ship, then it was time to really get to work. And we actually did a skin biopsy and other swabs and characterization and blood draws of the crew right away. So one of the things, the first things we looked at was what was the number of proteins we could see in plasma. You can see over here on the left. And what was the core number of proteins in the middle, you can see. And then what's the difference pre and post flight. So actually, as you can tell, the Venn diagram mostly overlaps, meaning that it's mostly the same before and after flight. Most proteins that are in plasma stay about the same, which is kind of good to see, good news. But there were some that changed slightly. You can see some were related to collagen genes, which we did see also as that change in the twin study. It's kind of a validation of that result. And what's interesting is some of the proteins it went down and have stayed down actually since the mission. So we're going to be keeping an eye on these to make sure that hopefully they come back or whatever they may be, that they are potentially just a response to the spaceflight the mission. But we did see some changes, and they basically are cellular stress in the immune system. If you look at what the changes, the changed proteins, this is what they're involved in. If you look at downregulated, it's mostly complement factors and blood clotting. So some of the pathways that we've seen before for other missions can indeed be modified and sort of perturbed here from spaceflight. Now, one other thing we want to look at also is not just what's in plasma, so that's just basically free floating in blood, but then also what's in exosomes, which are encapsulated small micelles in the blood that can carry DNA, RNA, proteins, and other messages. And you can see here they're implicated in a lot of different cellular processes, and they're about 40 to 160 nanometers. And so what we did is did proteomics on those as well. We could see the number of proteins per sample was less, and actually the overlap was distinct compared to plasma. So that means that each one of these layers of biology has a distinct profile. Then we did also what's the response to spaceflight, and here too we saw some different genes. So it's again a different biological response to spaceflight, which you can kind of see here. And then we also looked at the enrichments. If you take all those different proteins and think, well, what are they enriched for? You can see here that this includes chromatin assembly, chromatin remodeling, some defense response and immune response, and even telomere actually induced senescence. And what's interesting about that is we thought, well, the telomeres get longer here too? And it's a bit noisy, but on average, yes, actually they got a little bit longer from the blood collected in flight, which was kind of intriguing to see. The other few things we looked at in this mission was like, the microbiome. You think about your skin, if you're in a tight capsule, some people, your skin microbiomes will probably start to get pretty similar. We've seen this before, even with roller derby, this happens. And so we swabbed the 10 locations across the crew members and then characterized these data at a metagenome and metatranscriptome level. And indeed, we can see that, they have a lot of species per gigabase per region of the body. There's a lot of species to be found. But on average, the diversity decreased, meaning that the crew became more microbially similar to each other during and even after the mission. And if you're wondering, who changed, what are the species that changed? You can see it was an intriguing mix of some Colobacter and some species that are actually normally found in water, lakes, and streams, which is kind of intriguing to see that these are the ones that changed the most. So that was at the microbiome level. Then, as I promised, we also did a skin punch. So as soon as they landed, what happens in the skin? And this is using a genomics platform, which basically you can scan a light onto one part of the slide, cleave off these oligoconjugated antibodies, and then pull them out and sequence them. And that you've imaged these different areas of the tissues. So this is what it looks like. You can see just some crew members, 1 and 2. You can see here 3 and 4. And the staining here is for DNA or pan-CK, FAP, or asthma for vasculature. And you can see it's these beautiful images, but then if you want to quantify them and see what's changed, you want to look at differences pre versus post within the same person and then compare them. So we did that indeed. We could see all these different genes and really technically changing quite dramatically, and also across replicates. So you can see them consistently changing. And the ones that were changing the most were parasites and melanocytes. You can see the p-value at the top of what was the most post-flight dramatic change for these cell types. And so that was what we're thinking. I'm going to go back to this, what we're thinking for what is happening in the skin. We could also see macrophages change, meaning that the immune system even moves to some degree towards the surface of the skin, which may explain also what we saw for Scott Kelly, is that some of the disruptions in the immune system literally lead to a restructuring of how the cells look in the skin and where the immune cells are and how much inflammation that they might be bringing. So kind of interesting to see that as well. Okay, so some quick vignettes and highlights. So next, we are working with SpaceX for the Polaris Dawn mission, which is to repeat some of these measures for the next crew. Eventually, it'll be working with the Starship as well, so get this to work for this larger mission coming up soon. We also are actually planning a new twin study, which is trying to get a whole Starship full of twins over the next few years, and actually are working to make it so that this is a really ambitious mission that would go around the moon and eventually get to Mars with large sets of twins. So trying to increase the N, get more and more data, working with a number of partners at NASA, ESA, SpaceX, Axiom, and others to make this a reality. Okay, so that was the first half of this next decade, but then we also want to think about what might happen on cells on the way to space or boots on Mars. And also to put this in context, there's not just one space station that's going to be here in 5 years. There will be as many as 4 different space stations, which are currently funded or partnering with NASA. And we're not the only ones. So China actually wants to put crewed mission together by 2033, which is not that far. And they want us to get a sample return mission completed, where they bring samples back also by about 2030. So pretty impressive goals. And this gets to a question that is also high in the mind for JPL, Jet Propulsion Laboratory, where they want to do a Mars sample return mission and are planning on it. But so what's going to happen, though, is when you get these samples back from Mars, everyone wants to know, is there life on Mars? Did we find it? What does it look like? But you're going to pick it, do mass spec, do sequencing, do deep characterization, because this Perseverance rover, which landed on Mars last February, is in the middle of extracting samples and collecting air samples as we speak. And you can see here, there's the clean room where they build spacecraft, engineers putting together the final touches. And, engineers, they get a little excited sometimes. They might say, I'm sending this robot to another planet, so I'm going to take a selfie with this robot, because why not? And so that's very exciting. So actually, this robot's now tooling around Mars. This is a picture from the surface of Mars, looking, and here's Mars, looking in the soil that looks like this, collecting samples and then banking them and looking longingly back at Earth. That's what Earth looks like from Mars, waiting for them to get picked up. And so really kind of a poetic view of what is currently happening. But the big question we have is, what if we bring these samples back and we just sequence them and we just get the microbiome from these people who took a picture next to our spacecraft? So there's a lot of work to make sure that doesn't happen at NASA, what's called planetary protection. But it does beg the point that what we want is basically planetary genomics. We want to be able to track the presence and incidence and movement of any nucleic acid of any kind to see what it's doing, where it's going, and with whom it's sort of collaborating, if you will. And so to that end, this is kind of the visual to go back. That's one of the things, like if we want to know what's moving between these different planets, we need a way to track them and to monitor them. So we actually have a way that we've been doing that, and I'm going to tell you about it with a little bit of a story. So it starts about 8 years ago when my daughter, who's pictured here with an ice cream almost bigger than her head, was riding the subway and did at one point lick a subway pole while she was grabbing it. And I had this moment of complete parental terror, which was, oh my gosh, my daughter just licked a subway pole. What should I do? So I didn't know what to do. So I started to ask around, look around, and actually ended up saying, well, let's just start to collect data since we don't even know what's actually happening, what's in the world around us. So we actually went out and swabbed every single subway station in triplicate. You can see here's a picture of me swabbing. And, it's worth knowing that media is not the place for nuance, because I was asked, is it okay, Dr. Mason, I really want to know, is it okay to lick the subway pole? And I said, well, it depends on who's there and how long and a lot of factors. But probably fine in general, on average, writ large. And this is the only thing that got put for the headline. Most shockingly, this is also the picture that came with the headline, possibly the most disgusting picture I've ever seen. So in any case, you want to find out what's out there. You got to be careful what you say to people. But we also needed a coordinated global effort to really build this map, and not just do it for New York City, but to see what else is out there. So we started a nonprofit called Metagenomics of Subways and Urban Biomes, to go out and every day, once a year at least, map out the microbial members, sort of a microbial census for each one of these different metropoli, and put them together and share the data, methods, tools, and best practices and protocols. And what we've done so far is now over 30,000 samples collected around the world in this continued international effort to build a global metagenomic map. And you can see here some great pictures online of when people are doing swabbing and processing. And this data we published last year has led to a threefold increase in the size of the tree of life, including 11,000 new viruses. Most of them are phages, actually. So a really large addition to the field. And it's also built this kind of global map that I was alluding to, where we can actually have a K-mer for any sequence that's ever been observed, type it in, map it, and know exactly where it came from. And so this has led to a really unique view of where data is going and what it's doing. And so that's kind of the quick vignette. But now I wanted to think, switch gears a bit, because we're measuring biology we've been doing, really for centuries, millennia, really. But only recently have we been able to go from just measuring biology to start thinking about more actively defending biology, or even at least better understanding it. And so phase 3 is really where can we start to actually have armor, maybe like internal armor for human genomes, and just to think about off-target effects and to monitor them. And so the reason I bring this up is because we know that urine, even during flight, shows 8-oxoguanosine, which is a measure of damaged DNA. And you can see it spikes up for every single astronaut we've ever looked at. This is really a risky profile that's showing how much damage there is. And this is something, if you think about that, this was in low Earth orbit. This is in Scott Kelly got 146 millisieverts in 1 year in space. But if you go to Mars and back, it would probably be 1,200 millisieverts, and likely exceed the maximum lifetime exposure you're supposed to ever get if you compare this radiation dose to several others. And it begs questions then. Well, so we physically protect our astronauts, we pharmacologically protect them. Could we genetically protect them? Could we actually engineer a perfect astronaut? Or really, should we do such a thing? This is another big question, which we and others have published about. And it seems odd. It seems like a strange question, but it's worth noting that there's some ways that we know that you shouldn't do it. For example, secretly CRISPR embryos and then release them onto the world. That did not go well and was not at all a good way to do it. Conversely, for the Genome Project Right Consortium in New York City, we actually openly share methods, tools, protocols, our ideas, our plans for research, and get a lot of engagement and buy-in from the community before we do it. So this is something that we've been working on also more recently. And just to highlight the point that this is not an abstract and peculiar idea, CAR T trials, are visiting one of our Hangzhou sites, are ongoing, and working with them in our lab and others to modify T cells to cure cancer. So in particular, the way it works is a CAR T cells, you can take cells out of the body, you actually make a modified chimeric antigen receptor, or hence the CAR, and then grow those cells, reinfuse them back into the patient, and then ostensibly they get out and target exactly what is present on the epitope for the different cancer cells. Which usually can work really well if it's the right target, and sometimes doesn't quite work, but overall very exciting. Completely modified version of a cell and then put back into your body. And so this is not just 1 or 2 clinical trials. This is there's now dozens and dozens of clinical trials that are using in vivo editing, so using CRISPR or zinc finger nucleases in the body to make a desired impact of a gene modification, basically. And so again, this is not a rare or odd or occult event. This is state of the art and actually quite common. So with that as a backdrop, it's interesting. Well, could we add other genes? Could we basically take like a lesson from an elephant, which has 20 copies of P53 and has a lower cancer risk? So maybe that's one inspiration we could take, or take a look at tardigrades, what are called water bears, because they can actually survive in the vacuum of space. Look at them and say, okay, if it helps the human DNA withstand radiation and led to a 40% reduction in damage with just a temporary boost, what if it was permanently integrated into human cells? So you can see here, these are cells we've made in lab that have DSUP integrated into human cells, and that it's moving quickly to the site of double-strand breaks where these little speckles are showing up. And in particular, it led to an 80% reduction in damage that's dose-dependent from these hex cells with DSUP, basically taking and making a hybrid cell line that has bits from tardigrade and bits from human, as you can see right here, which is kind of intriguing to see. And that's just at the genetic level. So we also want to think about epigenetic ways to modify these cells in these populations. In particular, there's a wide range of tools with dCas9 or deactivated Cas9 that can go in and tweak a histone or tweak a methylation group. There's a really good review paper here about this. But this is something we've just started doing for targeting specific loci. And there's also something that DARPA has been doing, the preemptive expression of protective alleles and response elements, as a way to activate genes as you need them before you go into a dangerous zone. So really intriguing research here. So that's phase 3. Then, getting farther out into time and into the future, now we can start to test these genomes in space environments and start to think about other modifications where we'd need self-reliance for longer missions. We would need to have some other capacity just built into our bodies or with us. So one example I think about a lot is, and wrote about this in the book, is the gene for vitamin C. It's called GULO. The pseudogene is still inside of us. It's just so heavily mutated, it no longer transcribes that functionally or at all. But why not bring it back? If you think about this, instead of going to go get your vitamin C from mojitos, what if you could just make it naturally? And there's lots of mammals that already do this. So look at dogs and cats and bull. They make their own vitamin C. Rat, mice, squirrel, galago, lemur. The wet-nosed primates and mammals all make their own vitamin C, whereas some of the drier-nosed primates, such as ourselves, have lost this ability, which you can see here. So I think we could bring that back and make it so you don't get scurvy. You don't have to carry as much with you because you can auto-synthesize what you actually need in your body. So that's one idea. The other one is we have essentially amino acids that we need because our body doesn't make them again. So, well, what if we could have them? I think we shouldn't have to scrounge around the universe for amino acids if we could, in theory, make them. But for a long time, we didn't know exactly which pathways made which amino acid. And so it wasn't even possible to make it all in what's called a prototrophic cell. But now we know what they are. If you look here at these different pathways, this is how you could complete all the cycles from just 2 other species of our existing substrates in the human body and get it to work. Now, if that sounds simple, because biology is more complicated than this, but it does show that at least it's possible. And finally, we could prepare disease genes to lower the risk before someone goes into deep space. And this, too, has already happened in human embryos and has been completed. There's also other instances where you could modify a gene that's giving you a disease or change another gene. This is, if you're not familiar with the globin chains, like the alpha and beta globins, the globins among the most abundant proteins in your body and your plasma, unless you have a mutation or some challenge. For example, fetal hemoglobin, you can see here is in brown, goes away right before you're born, and alpha and beta that take over. But we know that, for example, you can target the specific repressor and actually reactivate fetal hemoglobin, which you can see down here in blue. And in many cases, it looks like it's curing the patient because you're modifying, what is, basically non-functional blood cells, and now suddenly you do. So really extraordinary clinical result from the past year one of my favorite papers here. And what this means is we're suddenly reaching a stage of cellular liberty. You're no longer beholden to the random genetic shuffle you got at birth. You can program your cells to do what you want them to do as you need them, and in different places and even in different planets. And so I think this concept of liberty and the ability to do what you want with your cells is very powerful, especially in the era in which we live, where these technologies are widely available. This then also lets it so you can have new intriguing structures in society. Like you could have 2 mom or 2 dad children, for example, that you could make. At least it's been shown here for mice. You can have 3-parent children. You could have a mitochondrial donor and a donor egg, and then have the father. So we know this is also already happened, is not only possible, but happened. You could even have, if someone had uterine cancer or had other problems conceiving, imagine a world where you have an exowomb or an extra uterine system that basically can keep you alive or even help you develop fully. And this is kind of a fascinating field because there's been some preliminary data that shows with this lamb, for example, it's growing in this kind of giant pouch that it mostly works pretty well. It wasn't all the way from beginning to end of a term, but it does show that it's possible to get this kind of exowomb created and utilized for a mammalian system. And so what that means is even from the very first cell, you have that cellular liberty. You can grow in any place you want as an embryo, as long as your parent takes you. You can modify cells to be functional or to resist cold. You do get a lot of options with these technologies. But the other place it goes that gets controversial is, okay, if we can do that, wouldn't we just do IVF and then pick the best embryo? And that's actually exactly what Orchid is offering as a company here. But it's been critiqued, I think, widely and mostly correctly, because can we really select the best embryos? Do we know how to do this? Is this something you can ethically do? And the way people describe it is, well, okay, here's what you do. So I think we can devise a metric, what's called a polygenic risk score, where you sum over all the different risk loci for a disease. Let's say schizophrenia, for example, and are you high risk or low risk? And what you do is basically just take a risk score distribution of cases and controls, look for the alleles that are giving you a high or low risk score. And then once you have that, then you can actually just optimize for the lowest risk embryo. And that seems reasonable, right? But who doesn't want a low risk embryo versus a high risk embryo? Except that you have to know what you're measuring. And so in particular, imperfect measures will give you imperfect metrics or sometimes wrong metrics. And in that case, this is exceedingly the case, where if you look at, say, just the risk alleles for, say, schizophrenia, you can see they're all over the map. And then what they ended up actually associating with most strongly was just the ancestry. What's the Black, white, Asian, Hispanic ancestry that was allocated in the survey. That alone tells you more than almost anything else that we've done. So it is early days for sure of processing the polygenic risk scores, but we know that we have to get a lot more data before we could truly, I think, utilize this for, say, schizophrenia risk or diabetes or any other disease. With that as a challenge, so we know it's going to be challenging. It's not there yet, but we do know that some of this can be done. We know that it is possible to have an idea of a directed evolution or a modified genome and do it just with breeding programs. This is actually what was done with the silver fox domestication experiment, which you can see here. And basically, a series of Russian geneticists got together in the 20s and 30s and said, oh, let's just take some of these foxes here. You can see, actually go back one that had, they let the more aggressive ones die of old age, and the young ones that were much more docile and playful, those are the ones that they then bred. And even within a few decades, the domesticated foxes started to have floppy ears, like a cute dog that they didn't have before. So it's an interesting change. And then they started to get juvenilized facial characteristics, because generally humans think that animals look very cute when they look like they're very smushed. So that's exactly what was done here, is to make this kind of, over time it became more juvenilized. I had a student in the lab, Lenore Pipe, who studied their brains as well, and some of them died of old age. We characterized the tame versus aggressive and saw differences in their brain chemistry. And so with that as an exemplar, we know it's possible to do a little bit of directed evolution, but the thing that I want to do is to make sure that we track it in terms of what it's doing to society, to the individual, what's the pleiotropy, the likeliness of success for risk score for any disease. And so this is a complex formula here, but just in the overall point is we need to include more than just does this drug work, or I think it's of course to most people here. Okay, so by 2060 we'd be getting pretty good at that, I imagine, and then we get to phase 5, which is where we could look at other planets and even synthesis of complete synthetic genomes. And so this too has some precedents already been done. The whole genome was recoded and plotted out. And then it starts to get kind of funky, where this is, I think, well, if I could have any kind of cell I want and start to add things, could I get some photosynthetic skin? I've thought about this since I was a kid, and I did the math one afternoon to say, okay, well, if I want to be by a pool and lay out my skin to basically hang out with everyone, how much green skin would I need to get enough energy? So this assumes that I'm spending, I've got basically a 300-fold epidermis expansion. I just want to try and do it in about an hour. And how many tennis courts would I need? I'd need about the equivalent of 1.96 tennis courts of my skin to be unfurled, to collect the sunlight, to be able to survive for the day and that amount of energy. So Fun fact, just kind of an interesting little calculation. But there, I think people often really critique these ideas because they say, well, you can't have like a half plant, half man kind of hybrid creature, tardigrade human. That just seems bizarre. But it's actually already happened many times with chimeric embryos from humans and pigs, other variations of them you can see here. And also, it's more common than you might think. Deli Ratifers are these kind of creatures that are called scandalous because they have life without sex, which is probably as awful as it sounds. But they basically survive on borrowed DNA, and they eat DNA to survive, and they can skip sex sometimes for a long, long period. And so this, seems weird until you look at humans too. We actually have about 8% of our genome is from an old virus, old retroviruses that are incorporated into our DNA. And so it's not just them. So, by that point, we'd start to reach out into just farther into the stars. But it begs the question of which stars and where would we go and what would survive there. So to take lessons in that regard, we have something called the Extreme Microbiome Project, which is looking at really unusual environments where microbes somehow still survive, which so far is basically everywhere on Earth. But we look at places that have really unusual fire profiles or salt profiles, salinity, radiation, different challenges that we can look at and then characterize them quite well. And as I alluded to before, there's another paper where we published ISS microbes and what they can do. I do have an expedition team field out this week, actually, out looking for deep sea vents that have a lot of carbon and also can have carbon sequestering and growing phytoplankton that can actually help potentially with climate change. You can see the collections getting down underwater. And this is Krista doing extractions right by where we just collected the samples. So this gets to the point where we have, say we collect a lot of extremophiles. Now, could we have them go to Mars or other planets, or would we modify them more so that we could have plants and animals that would survive on Mars, and it could maybe someday look like this on Mars, you can see here. And so the goal there is to really make it so by phase 7, we start to have Mars becoming self-reliant. They can actually, for the most part, control their own destiny, have their own resources, make their own biopolymers and therapies that are really on site and in situ. And this is something that we're also excited to start to see branch out, because once it does, then you have something that's basically called planetary liberty. You can then go between any planet you want. Right now, everyone has 0 planetary liberty. You can't go anywhere because we're on this planet. It is wonderful spaceship Earth, and there's a lot to do here, and it's very beautiful, but it's still only one place. And the liberty of planetary liberty is something we do not yet have the luxury of, but I hope someday will. And the reason is because it used to be that we had no planets to go to at all. So if you look before 89, there were basically no exoplanets found or planets that are outside of our solar system. But then in the mid 90s and especially in the early noughties, we started to really use the transiting method to find a lot of exoplanets, which actually when the planet goes in front of the sun, you can see kind of the blip of light because it's blocking between the sun and our telescopes. But here you can see over 4,000, almost 5,000 planets, I think, now have been discovered. And a lot of them are in the red zone, meaning it's the radial velocity. It's very simple in terms of being able to pick up changes there and the planets that you're picking up. And you get some characterization of them as well, which I think is very fascinating. But we have the biggest point, though, is it's not 10 or 20, there's thousands of them. And what's even more exciting is a good number of them are potentially habitable, meaning they might have liquid water. The gravity would be close enough that we could potentially get there and survive. It might be a little bit dicey, but they're certainly close enough that we now have candidates that are the best candidates to go towards. And you can see here, there's something called an Earth Similarity Index, or ESI, which is how close are some of these new planets to Earth. And in particular, we can see that they are, one is of course the same as Earth, so it's the closest as Earth. But if you look at things on this chart, you want to calculate the surface temperature, the density, the solar flux, and features of the planet, and make an ESI score. And when you do, you can see that there's other planets like Proxima Centauri b that have actually better profiles than Mars or the Moon, which here is kind of white. It's not even that good. It's less than 0.8 for the ESI. But if you zoom out a little bit more, you can actually see here, this is within 15 parsecs, you can see all these blue and purple planets that are nearby us that could be potential homes we could go hang out on. Here's about 20 parsecs. And if you zoom out a bit more, this is our galaxy looking at from the top or from the side on the left and the right here. And you can see this is every single exoplanet that's been found to date. And look at all the different spots of purple and blue. These are places that we could potentially go to, land maybe survive even today with much modification to our bodies or to the planet, but maybe just go on land. No, we have to get there, but it shows that there's all these planets that are pretty close to us, certainly within our galactic neighborhood and in our galaxy in this arm that are potentially new homes that we can and should look at. And so that leads to the last part of the talk, which I want to make is a bit more forward thinking and a bit even more than 500 years, but thinking long term. At this point, at the end of the 500-year plan, we could start to launch generation ships, which is where you'd have multiple generations live and die on the same spacecraft on their way towards one of these other planets. And now you think, well, that seems, if you're stuck on a spacecraft, does that seem cruel or unusual? Is that even, really legal? But you have to know that, I think it seemed to be impossible to stay in one place for a long time until the COVID pandemic hit, which now we've all gotten a little bit of experience doing that. But the challenge is also over time, would those humans feel themselves as the chosen ones, like the honored members of humanity that are leading the way, or would they become bitter and resentful and want to turn it around or scuttle the ship? We don't know. But one thing we do know is we'd have to give them a really good reason as to why. If there's really people going into a generation ship on their way to another planet, multiple generations living and dying, they would need a really good reason as to why. And so that's I think the biggest thing I want to close on is the bit of a philosophical piece is why do all this space research? Why do any of this at all? Because a lot of times people say, well, there's poverty, there are other societal issues, there are other things that need to be addressed. So why do all this? What's the big deal with space research or in general, some of these tools? And I can tell you exactly why. So in particular, if you look at this, for example, this headline, about 3 billion birds had vanished in the past like 50 years. And you can see on the right, some of the species are gone. Some have increased in their abundance, but most are gone, just depleted from the landscape, mostly because of habitat destruction. But we're losing species, and they're just disappearing, losing members of species, and we're losing species before we even found them. So a lot of microbes have also been lost, very likely, as far as we can tell, before we got to sequence them, because diversity is decreasing in almost every subject in some of these trials. And this is not the first time, right? There's been multiple times throughout human history and Earth history where there's been mass extinction. And so this is actually one of my favorite plots to show is, here's the last 500 million years of the Earth's temperature. You can see we've had a world with polar ice caps on the bottom and a world without polar ice caps. Sometimes almost all the ice on the Earth melted and then it refroze again. And you can see here, every time though there's been a dramatic change in the temperature, that is when we see a massive extinction. So in particular, you can see anywhere from 75% to 96% of all species just wiped out during some of these massive extinctions. So it's clearly there's a risk that's happening. And even right now, we're trying to avoid towards the end of this chart, point number 5, if it increases too fast because of, say, climate change, we might just create our own extinction. That's something we're trying to fastidiously avoid. But, as interesting, I think, as this chart is, I think it's even more interesting to take a step back and think, well, there were even times 700 million years ago where the whole Earth was basically a big snowball. It's called snowball Earth. It got that cold. And so the Earth has been through a lot of changes in cycles and temperature and structure and bombardment over the years. And if you take basically that 500 million years and you zoom out and then you put this in basically the history of the Earth and the likely future, you can see here that we're in a very nice, but a very temporary temperature canyon. This is a perfect spot where, the Earth is relatively stable, feels warm and cozy, but it wasn't always that way. You can see on the left when it first formed, and it won't always be that way. In about 4.5 billion years, the sun will become a red giant and engulf the inner planets. So even if we have world peace and everything's wonderful and it's kumbaya, if we're still here, we will all be burnt to a cinder on this planet. And so what this means, I think, is we have, think about what would what would happen, what would be at risk from all these different organisms. And more importantly, it wouldn't even be 4.5 billion years, because after about 1 billion years, the sun will get hot enough to start to boil the oceans. Actually, go back this really quickly, about 1 billion years, all the creatures on Earth, including consumers and producers, will all start to be basically boiled if they're still here. And actually, when I was writing this book, I was doing math and the calculations of when the temperature would increase to get too hot to survive on the surface. And I used to always think when I was a kid that I had 4.5 billion years till the sun got eaten till the sun eats the Earth. And I thought, well, we've got time. But then in doing the math in updating the book, it'll really be in about 1 billion years where the sun, it'll just get hot enough to start to literally boil the oceans. And so as I was writing this, and I went downstairs and kind of was moping around the kitchen. And then my wife said, you know, what's wrong? And I just looked at her and I said, I just thought we had more time, and explained the preposterousness of being worried about 5 billion versus 1 billion years, because 1 billion years is still a long time, is what she said. But then I said, yeah, but think if you thought you were going to live to be 100 years old, and someone walks into your room and says, no, that's it, you're going to be dead at 20, you would be upset. You would feel shocked. You feel like something had been taken away from you, that this plan and this vision of how long you could and should live was cut short. And that's exactly how I feel about our planet, is that we have an extraordinary diversity of life. We have, producers, consumers, decomposers, all these ecosystems, but it is at risk. And what's extraordinary about this picture, which you probably remember from elementary school, is this is kind of the ecosystem of the world, is that you can see that there's three kinds of creatures. And then three kinds of creatures, but this is not the only kind of creature that exists on Earth. We've always thought of this, that there's only three, but I argue to you today that there is a fourth and distinct kind of creature, which you can see here is basically us. There's only one kind of organism that is aware of the frailty of this ecosystem, which you could call the guardians, or you could say quite literally the guardians of the galaxy, but really only humans know about this, can only see this. Only humans are extinction aware, and therefore only humans can actually prevent extinction. And I think we have a duty to all life, present and future life, but even past life, towards the serving as shepherds of life, as serving the only ones that are aware of the frailty and can prevent it. So we're the only ones that can actually act on this duty, because we're the only ones that know it's there. It might be some other aliens in the universe somewhere, but to our knowledge, it's just us. And if that's true, that means we have a duty to all life, and potentially even extinct life, like Colossal Biosciences recently has begun to start to work on bringing back the woolly mammoth, and Revive and Restore projects working on species that are close to extinction. So we've actually been working, for example, with them on the black-footed ferret to see if we can take some of the last remaining members and clone them and bring them back to health, working with Revive and Restore and Ben Novak here, and really preserving the life that we have and even bringing back extinct life. Now, you have to do it carefully, because just as you take away a species from an ecosystem can have profound and devastating effects, adding one back in could do the same. So we have to do this slowly and carefully, of course. But it does give us, I think it really puts us in an exciting time for the field and for genetics, of what we can do and what we can think about, and what I think we have to do and should do, frankly. So, okay, so that's the view of humanity and what I think we should do. But the phase 10, the last phase, is the largest and last philosophical part, which is, okay, well, if we go to another star and you just keep doing, kind of iterate on this, you think we'll go to one star, go to another star, we'll have to keep hopping like locusts from one place to another place as stars start to engulf their planets. Eventually we're going to run out of places to go, right? Or you think long enough, what's going to happen in the universe? There's basically, one of three things that'll probably happen. There'll be either the big rip, or some amount of constant dark energy in the universe, or the big crunch. And so after the Big Bang, we'll either have everything collapse back into itself again, maybe have a new Big Bang, or the thing of universe will get so fast moving apart that nothing will be close to each other, and galaxies will become separated. And eventually the planets will all be too far apart from each other, and it'll be a cold, dark, and distinctly sad kind of universe. But we don't know which of these three it'll be, but it's interesting to think about it. If we knew it was any of them other than a constant universe, it begs the question of, well, would we try and modify the universe? For example, should we prevent this end of the universe? Or, I think it's the biggest and final ethical question, do we allow the destruction of the universe in the expectation that life would arise again in the next universe? Now, we don't know the answer, of course, because it's we don't know, as far as I know, it's the only universe we've had, but it could be another version, but it is the only one we know of, and the only one that we know that has life. And if that's true, I think what we do is do what we always do, is that we would just survive. We would probably try to restructure the universe itself to make sure that life would survive, which I think still matches our duty of what we should do, which raises interesting questions about the future, but I think very much what we, again, can and I think should do. So with that as a closing, I want to thank the twins investigators who are part of the twin study. This is the group of the gang of 10 that all worked on this, which is fabulous and collaborative and a great group with whom I still work with, most of them actually. I want to thank, of course, the astronauts who gave blood, sweat, and tears to some of these missions. This is the poster that they got to pick. You can see here Scott Kelly in the front. I also want to thank the current and past lab team members. This is a more recent snapshot of a lot of people on the team. And you can see here some of the older pictures of the team also that made all this work possible. And here people had been vaccinated, so they were a little bit happier, you can see here. And finally, of course, funding from NASA, from Trish, World Quant, NIH, XC, as well as the Gates Foundation, Sloan Foundation, and World Quant again. And then really all the collaborators, colleagues, teams around the world, friends who helped make this work possible. I can only talk to you here today because I have an amazing team that makes all this work possible. And I will close. My last slide is a quote from actually Ernest Shackleton. This is a picture that's up actually where the rockets are at Kennedy Space Center. And when Ernest Shackleton about 100 years ago wanted to get people to come with him to the Antarctic, to the South Pole, he'd say, okay, here's the advertisement: hazardous journey, small wages, bitter cold, long months of complete darkness, constant danger, safe return doubtful, but honor and recognition in case of success. And I think we're asking, again, a high price to pay and a high risk for all the brave astronauts and souls going up in private missions, government-sponsored missions to these places that are going to get farther and farther away from Earth and more dangerous. But I think it's an extraordinary time in space flight and in biology and in genomics, and we're really just at the beginning, I think, of this kind of launching stage where humanity can start to peer just past our own horizon towards other planets and actually reach for the stars. So with that, happy to stay and answer some questions, and thank you so much for your time.